we go. The different morphological states of every cell depends on the patterns of expression they are having. Once the pattern has started or selected to be different, we will experience a different phenotype of the same cell. It's not about changing everything. Sometimes it is just about the change in the pattern of expression of one gene, because that one gene will going to create a transcription factor, and that transcription factor will going to affect the expression of a category of genes. There will be several downstream genes will be affected because of that just one thing. That one stuff might be good enough to bring about a huge change. Such stuff, such thing, which is defining the phenotype of an organism or of a cell, the change of which alter the phenotype of the cell, such things can be considered as marker. Such things can be called or can usually be named as the markers, which are responsible for the phenotype or which are characteristic of the phenotype of a particular cell. Now, the hematopoietic stem cells, and you can see different cones and rods. You need to understand these are the same pictures. When a cone is placed like this, that means the expression of this thing, whatever is written here, is more at that time, and then it decreases from here to here. What is written here? The self-renewal, the ability to make more of the cells of its own type. The hematopoietic stem cells also require to produce the cells of their own type, ताकि वो maintain रखे ना एक certain number to produce all the cells for the rest of our lives. Not all of them can be converted into the other cells. So the self-renewal ability will going to be decreased once the cell is converted from one phase to another, from hematopoietic stem cells to become a multipotent stem cell. How we can distinguish at a protein level or at a gene level, or what is the marker for this conversion? The markers are written here: CK1T plus SCA-1 plus LIN minus. Whatever is written here means is indicating the state of the cell as hematopoietic stem cell. And when it is converted into a multipotent stem cells, that thing will become smaller. So once the expression of CK1T is started decreasing in the cell will be committed, or cell will started changing its phenotype from hematopoietic stem cells to become a multipotent stem cells. It's your task to see the individual markers and to read the properties of those individual markers, and you will come to know that how these individual marker gradually changes the phenotype of the cell. Once it becomes like hematopoietic stem cell, still it is capable of self-renewal, and after that it will produce two progenitor cells. They are called as committed precursors. It's still a general term, committed precursors. That means now they are destined to become a few type of cells. It is like somebody started a journey, and after some time it will become like two ways over there. Ek doraha hai over there. If the cell enters on one path, that cell will not going to be ended up in the same way like the other one. This is like a defining moment for both of the cells. Once the cell entered or selected one particular path based on the expression of certain genes, they will be called as committed cell. They will going to end up into a one particular type of cells. For example, if the cells are a progenitor cell like this one, that will going to produce beta cells and T cells only. From here on, this is a different path. This is a different journey. This needs different combination of genes. If this progenitor cells select that path, select that way, it will going to produce all of these cells present at the bottom. But they will not going to produce ever beta cells or T cells. It's a different path. Similarly, this path will not going to produce any of the cells like neutrophil, monocyte, eosinophils, basophils. They will not going to produce any of those cells. It's a different path. They will only going to produce the beta and the T cells. That's very important. So a committed cell is the one whose patterns of expression is changed. Now it is irreversible for that cell to go back. It will proceed further to the next journey, to the next path. Such cells are called as committed cells. So we have like two committed cells. Let's see how the pattern of expression changes in that journey. Most of the important markers are written here and their phenotypes are written here. Look at that. This is the cell division. This is the cell division. Can you see it is gradually increasing? So the hematopoietic stem cell, when start its journey, the ability of that cell to divide increases because it needs to produce more cells. It increases, goes to maximum and then decreases and then stop. Then they will not going to divide after that. They will become differentiated. This is written here, the lineage specific markers. Until at that level, the cells are somehow carrying the stemness. But after that, they will select their path. They will be on different journeys. They will be on different tracks. Whatever the markers they are carrying, those markers will define what will be their destiny. Whatever the markers they are carrying, proteins, transcription factors, they will define that what is the destiny of their journey. Their genes, what are the proteins they are expressing? कोई specific protein है या नहीं है जो मैंने बताया शुरू में markers की definition बताते हुए. Now सारा जोर ही मेरा markers के ऊपर है. Okay, clear करके जाना बिल्कुल 100%. That marker or that expression of one particular protein which push the cell to select a path and to end it up in a particular destination, that is called as lineage specific marker. That is the marker which take that cell to a particular destiny. मैं example दिखाता हूँ. पहले आप ये समझ लें, the lineage specific markers are not here. It is not here. It is absent. The cells can select any path. The select can go anywhere. Okay? Think like that. Stay like that. You can be anything from here on. वो कंग फू पांडा में कहते हैं ना, ये डिसाइड नहीं करता कि आपका पास्ट क्या है. Who you will be. इसका फैसला आपके पास से नहीं होता. इसका फैसला इस पास से होता है कि what you will going to do from today or from here on. There is no lineage specific marker here. That means the cells, they can select any path. And over the period of time, you can see the lineage specific markers increases. That means it is the cells are started, selected their journeys, selected their tracks, or eventually selected their destinations. आगे, for example, वैसे इनको भी पढ़ना चाहिए लेकिन आप वो पढ़ लेंगे same. For example, this progenitor cell will going to produce two progenitor cells. The one is called as pro beta cell, the other is called as pro T cells. The pro T cell will produce a pre T cells, and the pro beta cell will produce a pre beta cells. This phenomena is also called as lymphopoiesis because they are the lymphocytes, more accurately. इसलिए इसको lymphopoiesis भी कहते हैं. It is your job. If I am not telling you, if no instruction is telling, you should be curious. Which of the cell will become a pro beta cell, and what is the difference between the pro beta cell and the expression of the pro T cell? There should be a difference which defines that this one will going to become a pro beta cell, this one will going to be a pro T cell. It's your job to dig and find out the difference. Please keep that curiosity in your mind. Ye kaun hoga? And then eventually, once it is this one, now the pro-beta cell will never ended up into a T cell. It's a different track. Its destination is beta cell. Similarly, a pro-T cell will never ended up as being a beta cell. Its destination is a T cell. It will become like a pre-beta cell, the pro-beta cell, pre-beta cell, and then for further differentiation, they will become like a mature, differentiated version of the beta cell. Similarly, the pre-T cell will become like a mature T cell or a T cell. If you are working or writing or trying to think about something out of the box, if you want to think more, if somebody wants you to create proposals, to write things for them, and they are working on these areas, on these lines, these are the places where you should look at. If the textbook is not mentioning any specific markers or it's not blaming any one particular thing, then it is your job to go study and find the paper, the things into the paper, the people who are working with those cells. The people were trying to find the difference between these two cells, trying to find out the culprit behind these two tracks. Who pushed them to become a T cell? Who will push them to become a beta cell? If you know all these things, it will be much easier to control these things, to control the creation of these beautiful cells. Similarly, let's go back to this progenitor cell that will going to produce two types of progenitor committed precursor cells, the CFU and the CFU B, M and E. That will going to produce two different categories of cells. Look at that, there are two different categories. One category, once differentiated, they will produce neutrophil, monocyte and eosinophils. This category will going to produce basophil, platelets and erythrocyte. And there's a lot of associated expression of certain genes. I want you to look into what is CFU, what that CFU is. And this is like a defining moment for those cells, that whether they will become like these three cells or they will become like the other type of three cells. And also there should be a difference in the expression of all of these cells, because all these committed cells will become ended up into these six cells. They must have a different thing. I mean, this cell, the CFU-MG, has something special which will push that cell to become a platelet, not a eosinophil. So these are the very critical molecular information which you should carry. And that is called as the process of differentiation. And once the cell select a journey, it is just like that. If you need to go to another place, at first you will select like the two ways, become separate. यहाँ से हम इस्लामाबाद जा रहे हैं मिसाल के तौर पर, so we need to select first to go away. हम मुल्तान की तरफ नहीं जाएंगे. We will not going to go onto the Multan motorway. We will stay on the M2. And then after that we will not going to turn from Pindi Patiya से हम फैसलाबाद नहीं जाएंगे, हम इस सड़क के ऊपर रहेंगे. So इस तरह से selection करते करते eventually we ended up into a particular destination. This hematopoiesis is just a fraction, is carrying a fraction of complexity as compared to the process of development. Development में क्या हो रहा होगा, imagine करें, जहां पर एक सेल से all sorts of cells बनने हैं, और ना सिर्फ all sorts of cells will be created during the process of development, but also they will going to create tissues. The tissues will going to assemble together to make organs. The organs will connect together to make organ systems, and then all of these things should be connected together with the help of the nervous system to work in synchronization to execute the function, the beautiful thing which we call as life. So it's very, very wonderful phenomena to study, whether somebody asks you or not, whether it is part of any course or not, whether it is required or not. This is my suggestion, my advice to you, that please, please, please make sure that you people should study the procedure and stages and molecular mechanism associated with development, for sure. That will going to change your perspective a lot. Life sciences में जो भी चीज़ है वो वहाँ से आई है. I can tell you, all sorts of phenomena are covered in that thing. Okay. So that is the process of hematopoiesis. I believe it will going to help you. If you have any question, I am open to answer your question. And you can guess all these cells are present in the bloodstream as well as in the lymphatic system, which we